Good afternoon, everyone. My name is Stephen, and I'm going to discuss the drug amantadine and reflect on which ion channel this drug modulates and how. Additionally, I will discuss what this drug is used for in terms of treatment. Amantadine is an antiviral drug, typically used to treat influenza A. It is a acyclic compound in structure associated with an amino group. Absorbed orally, amantadine is recognized as a prophylactic drug used for influenza A virus. Additionally, it is also used in association with Parkinson's disease to delay the onset of symptoms. Amantadine classifies as an M2 protein inhibitor. It interferes with viral uncoating within infected viral cells affected by influenza. Additionally, amantadine mediates the proton equilibration of the intraluminal pH of the trans-Golgi network. Here is a short, colourful animation of influenza A in action.
Influenza infection. It might start with a sneeze, but the underlying infection can cause far worse. Hundreds of thousands of people every year die from severe influenza virus infection. Everything begins when the virus enters our airways. Here, influenza viruses specifically attach to the surface of the epithelial cells. The viral membrane envelope contains the neuraminidase protein Na, important for the efficient release of newly produced viruses. The M2 ion channel promotes viral structural changes during cellular entry, and the influenza hemagglutinin protein HA, the key player for viral internalization, which facilitates viral binding to sialic acid decorated receptors and subsequently viral fusion.
The M2 protein plays an important role in both the early and late replication cycles of the influenza A virus. The M2 proton channel maintains pH across the viral membrane during cell entry and across the trans-Golgi membrane of infected cells during viral maturation. Upon the arrival of the virus entering the host cell, endosomal acidification occurs, and as a result, low pH activates the M2 ion channel. Functionality of the M2 channel can be inhibited by antiviral drugs such as amantadine, which essentially block the virus from taking over the host cell. Amantadine binds to the transmembrane region, sterically blocking the channel. Viral uncoating is interfered when amantadine is introduced as a result of the inhibition of the M1 protein released by ribonuclear proteins, which essentially fail to enter the nucleus, normally in which viral replication and transcription occur. In the next slide, there's a short animation of 3D models on amantadine interacting with the M2 ion channel. You will notice the area of attachment located on the transmembrane domain. Shortly after this clip, additionally, this clip will show amantadine interacting with the M2 channel as well. Just take a close look in the slow motion. Figure 1 exhibits the activation of M2 ion channels facilitated by manipulated pH levels. Each channel, tested by Wang et al., demonstrated a reversible increase, while the levels of pH were compromised at 6.2. The amplitude of the current increased monotonically with decreasing pH. With regards to treatment, amantadine is used as an antiviral drug for the treatment of influenza A, and its derivative rimantadine is looked upon more favorable as a result of its less adverse side effects associated. Additionally, amantadine is also used as a key mediator in the delay of symptoms associated with Parkinson's disease. Although it is not clear, amantadine may cause greater amounts of dopamine to be released into the brain and may block receptors for acetylcholine, the neurotransmitter. Amantadine is also purported to be used as a treatment for patients suffering fatigue associated with multiple sclerosis, as well as patients suffering from severe traumatic brain injury. Furthermore, amantadine is also used for the treatment of severe symptoms associated with cocaine-dependent patients. In today's age, amantadine and its derivative, rimantadine, is no longer recommended for the treatment of influenza A virus within the United States, as a result of newly emerged resistant mutations of the influenza A virus, all of which are 100% immune to amantadine treatment. Furthermore, in 2006, The Cochrane review concluded that the drug amantadine revealed inadequate evidence of its safety and efficacy through several randomized controlled trials of amantadine. To reflect on what we've discussed, and to highlight key points, amantadine is an antiviral drug used for the treatment of influenza A. Additionally, amantadine has been used for the treatment of the early stages of symptoms associated with Parkinson's disease. M2 ion channel activity is facilitated upon the transport of the M2 protein via the exocytic pathway. These are key points. The M2 ion channel facilitates the dissipation of all over the pH gradient across the membrane within the Golgi apparatus. Amantadine classifies as an M2 inhibitor, mediating the inhibition of the early stages of viral encoding within endosomes of influenza A virus, of which have not mutated into the resistant strains. Amantadine is ineffective against the treatment for influenza B due to the associated unique characteristics of the M2 protein. In contrast to this, further research into the effectiveness of amantadine is imperative, particularly to develop an enhanced understanding of the drug's ion channel interaction and activity within the virus. Quantifying a more effective drug, particularly with an emphasis on how drug resistance occurs, highlighting amino acid substitution, will enhance the development of better, more efficient drugs to be used in the future. Thank you very much for listening to my speech.